Thank you, Christos, for the kind introduction. Can you all hear me?
Sure, Christina,
Yeah.
go ahead.
Great. So yeah, as you said, I will present my recent experience of the short-term scientific mission. I had the chance to spend 5 weeks at the Department of Biotechnology and Life Sciences at the University of Insubria in Italy, and I had this chance through GIN-TONIC. So starting with the facilities that I found there. The Department of Biotechnology and Life Sciences at the University of Insubria in Italy. They work with many insect species, but they do focus on the insect species Hermetia illucens, and that was the species that we worked on. I have added here a few photos from the rearing facilities because I found, apart from the experiments that we conducted and was really interesting, first of all, I found really interesting to see how another group works with this insect, how do they handle it. And what I saw was that, let's say at the photo on the upper left, for the love gates, they use also for moisture source, but also nutrient source for the adults. They use bananas, and that was something new that we don't do in our lab. And it was they show me how they rear the larvae. It is shown in the second photo, and also what to do with the eggs after they were collecting it. They have standardized the specific amount of eggs, 1 or 2 grams, if I remember correctly, and they add it in a petri dish without touching the moisture source, the feed. But the newly hatched larvae are able to crawl and find the feeding source, as you see in the photo on the bottom in the petri dish. So starting explaining what I did during the 5 weeks of my stay in Italy. Before going there, we together with my professor, but also with Professor Tetamanti and Daniele, we discussed on the protocol that we will run there. And the protocol was related to include feed additives derived from microalgae in different percentages and formulate diets for for the larval states of Hermetia illucens. So from the time that I departed, we started formulating the different diets. And after the formulation, we started the rearing trials. And at the end of the rearing trials, we continued with the step 3 and 4, which were for which I had no experience before, about the immune response of the larvae and also how to handle the gut lumen of the larvae. I say again, I had no experience on this, and it was really fascinating for me. And that was the reason that I chose to go to this laboratory, because the researchers there are experts on this. So let's dive a little bit deeper on what we did. Starting with the diet formulation, we utilized the microalgae species Chlorella vulgaris, and we incorporated it in 3 different percentages. The lowest percentage of insertion was 1.5% of chlorella, and we reached up until 10% inclusion. And the bioassay was conducted in plastic cylindrical vials in 6 replicates, and we utilized about 30 larvae in its experimental unit, in its vial. The first recordings, the first data that we collected was related to larval growth and survival. Simply what we did every 2 days, we were taking out the larvae from their feeding substrate, cleaning them, weighting them one by one, and counting their number in order to create the growth curves of weight and survival, which I will show it to you on the next slides. The next step, the part that was with which I was not familiar at all, was the evaluation of the immune response of the larval hemolymph. And I will not dive very deep on these procedures, but I want to say a few words on how we did the experiments. For this part, we utilized larvae at their last larval stage. We excluded them from their feeding substrate, cleaned them in order not to exist debris on them, but we also used bleach and ethanol and sterile water, so in order to avoid the infection of other microorganisms. Then, after we have cleaned the larvae that you see on the upper left photo, we were utilizing a needle, a sterile needle, and we were, with this technique, we were able to collect the hemolymph of the larvae. The hemolymph of the larvae was the material that we would work on the next step. And after the collection of the hemolymph, we centrifuged them to separate it from the hemocytes. So after the centrifuge, we utilized the hemolymph and we mixed it with, we infected it with two different bacteria. As you can see here, we utilized the bacteria Escherichia coli and the bacteria Micrococcus luteus. Then, after we created a mix, and the preparation of bacteria, you can see this preparation in the second photo. After that, we have created this mix. The bacteria needed to interact with the hemolymph. So the researchers there follow a protocol which allows 3 hours in an incubator chamber at 37 degrees for the interactions of the hemolymph with the bacteria. So we left those mixtures for 3 hours in the incubation chamber, and then we took out the mixtures and created serial dilutions in order to alternate, in order to dilute the concentration of bacteria. And then we inserted, as you can see on the last photo, on the fourth photo, we inserted, we put it drops on agar plates to leave the bacteria grow. These agar plates were also incubated in the same chamber at 37 degrees, and the time for incubation was 1 night. So overnight, we left them the colonies to grow. Going with the next day, we were taking out the agar plates, and as you can see in all of the photos, the colonies of the bacteria grew. And what we had to do is to count the colonies in its agar plate and translate this data to the percentage of infection. So starting with the graphs regarding the larval growth and survival, on the left graph, it is apparent that the individual larval weight did not show differences, statistical significant differences among the 4 treatments. However, I would say that this is not a negative result because, first of all, the control diet was chicken feed, so it's a very nutritious diet. And also another outcome is that eventually the incorporation of feed additives derived from microalgae do not affect negatively the weight of the larvae. The same pattern we see for larval survival on the right graph, for which, as you can see, there were also notes that maybe in the graph it is apparent that there were differences in larval survival, but when the statistical analysis was conducted, no statistical differences were apparent. So both larval weight and survival were not negatively affected by the inclusion of microalgae in the diets. Continuing with the results that we received from the immune response, This was also the first time that I was handling this kind of data, but I found it also very interesting. And the people there really helped me to handle the data and to create the graphs and show me how to calculate the colonies and everything. So regarding the Escherichia coli, these are the graphs for the hemolymph of larvae infected with Escherichia coli. As you can see from the graphs, there were no statistical differences regardless of the feeding substrate on which larvae grew. So both control and feeds that contained microalgae were not able to suppress the bacteria Escherichia coli. But the results for Micrococcus luteus are more promising and more interesting because, as you can see here, all the larvae the hemolymph from all larvae reared on diets contained the Chlorella vulgaris, the microalgae, managed to suppress at some point the colonies of Micrococcus luteus. The highest suppression was recorded for the hemolymph derived from the larvae reared on 1.5% of Chlorella vulgaris, and yeah, this is a really promising aspect. So these were the data that I collected, but I did not only do that during my 5 weeks there. Another technique that I was very keen to learn was how to dissect the Hermetia illucens larvae, and also how to be able to extract the guts intact without breaking it, and how to collect the lumen in the gut. So after many trials, not a few trials, after many trials, because it's a difficult technique, I managed to dissect the larvae and collect the gut, as you can see in the photo at the bottom. This technique is used when the bioassays target on enzymatic analysis of the gut of the Hermetia. If some of you, like me, are not familiar with these bioassays, there is this interesting publication of Bruno et al. At which the researchers utilize pigment to show the different regions of the gut. And as far as I learned during my stay there, it is very important to know which region you would need for the different bioassays, because different regions can show different results and also have different pH, as you can see in the third photo. So yeah, it's very helpful to read this publication. These two photos are from my bioassays. On the left side, it is apparent that the gut has an orange yellowish color. It is the control color that someone would see if the larvae were reared in controlled diets, in chicken feed specifically. But the larvae that were reared in 10% of microalgae managed to alternate the color of the gut. So it is apparent that the gut on the right side, on the right photo, is more greenish and totally different in color from the left side. So in general, through these bioassays, we saw that microalgae are able to act like green shields, I would say, under certain circumstances against certain bacteria, and to enhance the immune response of the hemolymph of the larvae. More specifically, the Micrococcus luteus belong to gram-positive bacteria, and other studies has also shown that these specific gram-positive bacteria, where the hemolymph of Hermetia illucens is more prone, is more easy to mitigate them. While on the other side, Escherichia coli belongs to gram-negative bacteria, and other publications also show that the hemolymph on its own is not able to reduce the colonies. Of course, many ideas came in our mind after these bioassays, and we want to collaborate further. Why not to evaluate these diets also in another insect species, the Nibrio molitor, on which I conduct my PhD, so I have more experience on how to handle it. Also, the inclusion of microalgae as feed additives, they might have a positive effect on poor nutritional substrates, because we utilize these microalgae with chicken feed, so with a highly nutritious substrate. But what will happen if we combine it with other poor nutritional substrates? Will it enhance the immunity? Will it enhance the growth of larvae? And finally, why not to utilize byproducts from the production of microalgae, because microalgae is also an alternative protein and produced industrially. So they also produce byproducts, and why not to reinsert them in the food chain by combining these byproducts in Hermetia illucens diets. Additional analysis that we want to do, because we saw interesting results and we wanted to take it further, is to evaluate the lipid content of the larvae that were reared in those different diets containing microalgae, and also to evaluate the fatty acid profile of those larvae, which seems very promising. Apart from all the bioassays, we had the chance, we were very happy that the team from the University of Turin visited us. Sarah, Andrea, Laura, and Ilaria and Zaira visited us, and they wanted also to see firsthand how to dissect larvae. So it was a nice gathering there. Thank you guys for coming. And yeah, thank you all for listening me, and huge thanks to Professor Tetamanti, Daniele, and Martina, who show me all these new techniques and gave the time to show all of it. Thank you.